Once again, welcome to my lecture on livestock products technology in YouTube channel, that is Professor LPT alias Food Technology. Today's topic is milk collection, testing and reception. As you know, earlier I was talking about the milk related and milk processing topics. In between I have switched over to the animal byproducts. Now again I am resuming the topics on milk. So earlier I have discussed about the transportation of milk and related microbial quality, the factors affecting microbial quality during transport and storage and all that. Now I am in that continuation today going to talk about the collection or procurement of milk, that includes the milk collection center and related equipments and operations, and then testing of milk, for which different platform tests and their equipments required and procedures, and finally about the milk reception. Now let us first understand about milk procurement and transportation system. For most of the dairy plants in India, milk procurement is organized based on famous Anand pattern or Amul. As you know, it is a place in Gujarat that was one of the most successful dairy development program. Anand Milk Union Limited, it is popular brand as Amul. This pattern lays a solid foundation on which the infrastructure of village level, that is dairy cooperative society, then at district level district milk unions, and at state level milk federation has been built. So this is a system. First it was started in the Koira district in Gujarat, that is the latter named as Anand pattern, that became a model, and that was replicated in every district and in the throughout country in India, and it is one of the most popular successful dairy development program which is even followed in other countries. The system is still sustainable even today, mainly due to appropriate milk procurement system, which is the lifeline of dairying. So this is a very viable and suitable system for milk procurement for all dairy plants and then further processing and marketing. First, let us understand about organizing milk collection centers or MCC. So this is about collecting the milk from the farmers to a particular place. From here it goes to the milk chilling center or milk processing center dairy plant. So for this, how to organize a milk collection center? A systematic approach starts with a village survey to estimate marketable surplus milk and find out the potential for future growth in milk production apart from connectivity and road conditions. So we need to find out what are the villages where there is surplus milk and about their connectivity and road. And then similar surveys are conducted in all potential villages coming into that main road or secondary roads across length and breadth of that region where this operation should be done. And after establishing viability in a series of villages, a milk collection route is designed. So first we need to ascertain the viability, the available milk, the surplus milk from the dairy farmers, and then we have to establish the road connectivity and we should make a route. That is how a milk collection center should be established. Now about designing milk collection route. Once we have made a survey and planned to start a milk collection center based on the potential, we need to design a milk collection route, for which we have to see the number of villages in the route. Secondly, the quantity of milk available per shift. Generally it is taken in first shift, second shift, or morning shift and evening shift. That is the milk collection system. And then we have to see the distance from the dairy plant, that is the collection center or production center, farmers, and the distance to dairy plant. If the distance is too much, then we need to have separate chilling center before that. Then the distance from village to village, that is between the village, what is the distance, and another is the road condition, based on which only the vehicle can bring the milk. And finally, the time taken for milk transportation from the first pickup, the first collection point to the dairy plant, how much time it takes. So this time should not be more than 4 or 4.5 hours. If it is more than that, then milk will be spoiled. So based on all these factors, we need to design a milk collection route. In continuation about designing the milk collection route, the milk collection routes have to be given a time schedule so that the route vehicle reaches the dairy dock, that is the reception point in the dairy plant, at the interval of every 20 minutes. That is, there are many different routes from which the milk will come to the dairy plant. So this milk carrying this vehicle, it should reach at least with a 20-minute interval, not before that, because that much time is required for receiving the milk. So all the vehicles should report in such a fashion that milk reception is over within the stipulated time. So milk comes with the milk carrier from different places. It should be collected, platform test should be done and received. So that should be over, and then only the next one comes. So there should be a synchronization. So this has to be taken care of while designing the milk collection route. Now we will discuss about the operation of milk collection center, MCC. While organizing milk collection route, the perishable nature of milk, its microbiology, biochemistry shall have to be kept in mind. So when we are planning the transportation route or collection route, we have to keep in mind this, that milk is very perishable. It should reach to the place by 3 or 4 hours. And the microbial level and the biochemical condition we have to keep in mind. At each MCC, certain disciplines shall have to be enforced by organoleptic testing, wherein stale, sour, adulterated milk shall be rejected. So organoleptic testing, that is done at the collection point, milk collection center, in which some of the conditions can be detected and rejected, especially the staleness or sourness. By putting in mouth, immediately we can judge it. That needs some expert to understand. And then the dairy should always procure milk in both the shifts, as I told earlier, that is the morning and evening shift. Unless cooled in bulk cooler at the MCC, milk should be transported to the dairy in each shift. So sometime there will be bulk cooling tank in the milk collection center itself. So if it is cooled, then there is no hurry to transport. It can be done one day later also. If there is no cooling facility, then it should be shifted both morning one time and the evening one time, so that the milk should never be there more than 3 or 4 hours without cooling. In continuation about operation of milk collection center, the farmers should be trained to bring milk in clean vessels, because these vessels becomes the source of contamination for the raw milk. And the milk cans at MCC should be washed with liquid cleaner or washing soda. So within the milk collection center, the milk cans should be properly washed and cleaned or sanitized. At MCC, milk samples should be tested for fat content and solids not fat calculation. So this is most important criteria for pricing of milk. So in this milk collection center, the fat content is tested and also the solid not fat content is calculated. This is very important operation for payment to the farmers. And then a person should be trained for this purpose, which needs further supervision. So to do this test, even previously I told organoleptic test, and now this fat test, it has to be done by trained person, and there has to be proper supervision, because it is directly related to the price of milk and the profit to the farmers. All the glassware used for milk testing along with sample bottles needs to be washed after every shift. So these glasswares which are used for testing of fat, etc., it should be washed in the morning shift before using in the evening shift. So now we will see some of the equipment for milk collection. Number one is milk collection tray, on which the milk will be poured before it is going to the collection tank, and that is possible only after the milk is checked for organoleptic and other tests. Then there is milk strainer, that is a muslin cloth that will be kept on which the milk will be poured to reach the milk collection chamber. Then there is milk sampler. We have to take the milk sample for further test like we are going to discuss later. And then sample bottle. So after taking the sample, it will be kept in the bottle and it will be properly leveled. There will be sample bottle tray. There is milk measures like different capacity, 2 liter, 1 liter, half liter or 0.2 liter, etc. Then there is plunger. It's a very big rod with a circle of metal plate having holes. So this is basically used for mixing the milk. Even if it is a milk can, we can mix it, then only we can take. Otherwise, if we take from the top, that sample will not be the good sample. It should be thoroughly mixed, then only we can get the proper representative sample. And then aluminum alloy or plastic milk can. So milk can is very, very important for handling milk or collecting or transporting milk, as we know. And the plastic bucket and mug. So sometimes this bucket and mug is useful for different purpose during the milk collection operation. So already we have discussed basics about milk collection center and how it operates. Now we will discuss about the testing and reception of raw milk. Milk cans are loaded on conveyor in a particular sequence and each can is inspected for abnormal taste, smell, color. So when it comes through a different source, so the cans are kept in a sequence and it can go through conveyor, and then by opening the can there should be a organoleptic test to understand that any abnormality on taste, smell and color. And after that a sample is taken quickly to check the clot on boiling test. So if there is a lot of acidity, then it will curdle and we can understand that milk is not good. And followed by there is some other samples are taken for further laboratory test like MBRT and other bacterial load, which I will tell later. So this is done on a sequence again as per the collection center. In that proper order this is done. Now in continuation about the testing and reception of raw milk. So these samples I told earlier we collected. These samples are checked for titratable acidity, MBRT, that is methylene blue reduction test, that gives a rough idea about the microbial load, and for adulterants. So nowadays it is a very common problem of different kind of adulterants like sugar, starch, urea, soda, water and preservatives. As you know, to make it thick they use starch or urea or sugar to make it or sometimes they use soda to neutralize the acidity, or they add water or different preservatives. So all these things we need to check later in the laboratory. If acidity in the collected milk is more than 0.15%, that is expressed in lactic acid, it should be treated as sour milk. So milk which is more than 0.15, it is not very suitable for heat processing. And then MBRT, as I said, methylene blue reduction test, it gives a rough idea about the microbial load, and at the time of reception it should not be less than 30 minutes. So this is the time required for the bacteria to grow and make a color change. So if it is taking less than 30 minutes means it is very heavily contaminated. The bacterial load is very high. If it is 1 hour or more than 1 hour or 2 hours, then it is very good quality milk. Then milk failing in organoleptic acceptance is to be treated as sour milk. As I told, first there is organoleptic test followed by clot on boiling. After that the other laboratory test. So during organoleptic testing itself we can understand any bad odor or even sourness if the acidity is high. So such milk should be considered as sour and it cannot go for heat processing at least. Here we will see some of the important equipments for milk testing. Particularly here we are seeing mostly for the fat test, that is Garber centrifuge, butyrometer stand, butyrometer shaking rack. These are all required for fat test. Lactometer with jar and thermometer. These are all for measuring the specific gravity or SNF. Then we need milk pipettes, bottle cleaning brush, pipette cleaning brush, butyrometer cleaning brush. Acid bottle or alcohol bottle, these are required for fat test. Then lock stoppers also required for that. even cans with acid and alcohol. In addition, we need separate facilities for MBRT test or other microbial test that we will see later. Now briefly we will discuss about MBRT, that is the methylene blue reduction time, that is an indicator about the microbial quality of raw milk. So this will be discussed more in practical classes. So indirectly it shows microbial load. If the time taken for this test is less, that means there is very high microbial load. So in this some sample is taken in the milk test tube as you see in the diagram in the picture, and there a dye is added, that is the methylene blue, at a very diluted level, and then it becomes blue. Now if we keep it in incubation around 30-37 degrees Celsius, we have to wait for the change of color. So if this color change takes 5 and above hours, then it is a very good milk. That means the microbial load is very less. If it is 3 and above hours, then it is good. When 1 to 2 hours, it is fair. And when it is 30 minutes or less, it is poor quality milk. Means it has got very high bacterial load. So the reduction in color is very fast. So this is how it helps in quickly understanding the microbial load of the raw milk. Here let us understand the bacteriological standard of raw milk. There is a BIS standard IS 1479. So here actually the total count or standard plate count per ml is estimated, which we will learn details in the practical class. Sometimes it is also called total viable count. So here the milk will be diluted and it will be taken into the proper medium, and then it will be incubated and hold for minimum 24 hours, and then we can count the colonies. From that we can estimate the number of organisms per ml of the milk sample. So when this number, that is the microorganism, is within 10 power 5 or 2 into 10 power 5, then it is very good, that is per ml. When it is 2 into 10 power 5 and up to 10 power 6, it is good. And when it is 10 power 6 to 5 into 10 power 6, then it is fair. And when it exceeds 5 into 10 power 6 or even touching 10 power 7, then it is poor quality. So the pasteurized milk should have a SPC per ml about 30,000 per ml organism in pasteurized milk. Then only it is a good quality or proper pasteurized. There are certain platform tests, that is the tests which are done during the milk reception in the platform nearby. That's why they are called platform test, in which first is the organoleptic test, followed by that is COB, clot on boiling, or alcohol test, sometime MBRT or research urine test similar to MBRT, or sediment test. So in case of sediment test, then we have to see how much is the sediment coming after holding for some time. So if it is 0, then it is excellent. If it is 0.2, good. If it is 0.5, it is fair. If it is 1, that is in mg, then it is poor. And if it is 2, it is very poor. So this is another test. There are few tests, as I said. Those are the platform test, which is done very quickly during the reception platform, and that's why they are called platform test. So we are talking about the milk collection, testing or reception, so in which the record keeping is extremely important. So here we should know some of the registers for milk collection. Of course, these are old system. Nowadays everything is computerized, it can be programmed and everything can be entered into the computer also. As per earlier system, following is the register like passbooks or monthly cards for dairy farmers, because there has to be proper record from the dairy farmers. Then purchase registers, testing notebook, which will record all the tests done for each sample and their result. Payment register for payment to the farmers or producers. Then members register, cash book, general ledger, and dead stock register. So all these are the standard procedure for recording everything for a milk collection center or milk cooperatives. But nowadays it has been computerized and it is made much more easy by entering everything in a very proper software or apps. This is now about bulk milk cooling tank. This is one of the important requirement in modern milk collection center. So most of the milk collection centers should have the bulk cooling tank. These tanks can cool milk from 30 degree to 4 degree in 3 hours. The tanks are available in different size like 250 liter, 500, 2000, 300, 3000, 4000, even 5000 liter capacity. So after collection, the milk can be cooled here itself. So there is no urgency to transport the milk to the dairy plant immediately. The integral condensing unit is hermetically sealed and use R22 refrigerant. So there are specific refrigerant for different cooling purposes, or different refrigerants can be used. So these are built in stainless steel and with agitator assembly. So these containers are most hygienic. That's why it should be stainless steel container, and there will be agitator inside for mixing. There will be on off switches for agitator and cooling, and digital display of temperature. So there will be a display about the cooling, how the temperature is going down and what is the final temperature. So this is briefly about the cooling tank or bulk cooling tank in the milk collection center. Here we will see some special caution in milk reception. So far what we are talking in general for milk reception, which will mostly go as a liquid milk processing, or we call market milk or some of the other products. But in case of making certain special products, the milk quality should be very special. So milk procurement for a specialized dairy product like cheese requires a high level of scientific approach. Milk required for cheese production has to be wholesome with its natural contents like fat, protein, minerals present in a particular species milk like cow or buffalo. The milk should be proper with natural composition of the milk. Then abnormal milk due to mastitis. As we all know, due to mastitis, the milk will be different. There will be high somatic cell count, even sometime pH will be higher, or milk with rich in colostrum, or due to the late lactation is not acceptable for this special kind of products. And milk has to be free from off flavors, free fatty acids, residual antibiotics, because that will prevent the growth of culture. Traces of detergents, sanitizers, pesticides or insecticides, and sometimes aflatoxins, mycotoxins or other heavy metals. So all these things should be free. The milk should be free from all these things which cannot be used for special kind of products like cheese. Now there are certain modern machineries, equipments for milk testing. We have already discussed about the milk testing in a traditional way. There are certain modern machines in the big milk industry which can perform very quick in very short time. So one is electronic milk tester, or sometimes called electronic milk tester. It requires very small volume of milk sample and can perform 120 to 150 fat test per hour. That is 120 to 150 samples per hour with auto zero facility. Its fat measuring range is from 0 to 13%. So the sample which has got fat at the range of 0 to 13% it can measure. Another is electronic SNF tester. So here it can perform 100 tests per hour with instant digital display without the help of a chart or table. So normally for SNF we have to use a chart and for correction of temperature and calculation etc. But here it is a sensor based, so it can do directly and very quickly. So there is no requirement of any chemical, and it is microprocessor based. So the SNF measurement range here is 0 to 12%. So in continuation to modern milk testing equipments, so here it is ultrasonic SNF tester. So previously we saw electronic SNF tester. Here it is ultrasonic SNF tester. It is a portable with a measuring range of 0 to 20%, that is the SNF percent, and suitable for all types of milk. It can test 80 samples per hour with digital display of the results. Similarly, there is another one, that is portable milk analyzer. So which can measure all the parameters in milk. So it is designed to measure fat within a range of 0.5 to 12%, SNF 6 to 12%, protein 2 to 5%, and corrected lactometer reading 20 to 35, and added water 0 to 6% in a milk sample within a minute. So if we just give the sample of milk, it will tell all this, that is fat, protein, CLR and water. Here no chemical is required and it is suitable for all milk like cow, buffalo or other mixed milk. So now we are at the end of today's lecture. Our topic today was the collection and testing and reception of raw milk. So we have discussed about the collection or procurement, about the cooperative structure, then the milk collection center, what are the requirements and systems for establishing or operating milk collection center or milk collection route, some of the important testing of milk and how the milk is finally received and cooled or bulk cooling, etc., and the other requirements, equipments, registers, record keeping, and some of the modern milk testing facilities or machineries. So all these things we have discussed. Further details you can learn from the proper textbook. Thank you.